Potato osmometer experiment aim to study the process of osmosis by potato osmometer. Requirement: fresh large size potato, peeler, 10 to 20% sucrose or NaCl solution, petri dish, beaker, water, pins and the knife. Osmosis is the movement of water from its high concentration to its low concentration. That means water is going to move from a dilute solution towards concentrated solution area. And that's what we are going to perform the practical. Let's understand. You can see as one side there is a high concentration of Na plus and at another side there is Na plus in the low concentration. That means at the high concentration area water is low and in the low concentration area the water is high. So water will try to move from low concentration to high concentration area of Na plus. Let's see the figure what we are saying, from dilute solution to concentrated solution. So you can see the water will move from low concentration area to high concentrated area. The movement of water across the semi-permeable membrane will continue till it is under the equilibrium. When at both sides the concentration gets equal, this movement of the water will get stopped. Osmosis is of two types, exoosmosis and endoosmosis. Exoosmosis: when we put the cell into the solution whose solute concentration is more than the cell sep, that is called hypertonic solution, water comes out of it. That means the cell sep is having the low concentration and the outside solution is having higher concentration. Let's understand. Inside the cell there is low Na plus and outside there is high Na plus. So water is going to come out of the cell to compensate the concentrations of Na plus. This is called exoosmosis. In exoosmosis, it is leading to the cell cytoplasm shrinkage. So that is also called as plasmolysis. We are going to see into another practical. Endosmosis: when we put the cell into the solution whose solute concentration is less than the cell, that's called hypotonic solution. Here water will flow inside the cell, due to which cell will swell up and become turgid. Here hypotonic solution is a simple water. For example, we put a cell or a resin into simple water, due to which water get soaked by the cell or the grape. Let's make the setup. As you can see in the image, a petri plate is there. Inside simple water is filled, where the potato with the cavity is put, and inside the cavity we have put the NaCl solution. You can see the image very carefully and make the setup as per this image. You can see in the petri plate there is a low concentration and inside the potato there is high concentration, which is NaCl solution. So as we can see, water will enter inside the cavity of the potato because the concentration is high inside the potato cavity as compared to outside petri plate solution. Let's do the experiment. This is the beaker with the simple water. This is petri dish. This is a conical flask full of NaCl solution, and this is potato with cavity. filled and properly ready. Let's put it into the Petri plate and pour NaCl solution in the cavity of potato. Now add a drop of safranin so that the water is visible. It's a colorful, otherwise we won't be able to see its level. Now take a rod or any other material and measure the height of water into potato cavity. Now I'm going to dip this inside the cavity and let's just see. Now we can see the level, it's little bit colorful. I can observe, you can see in the image where, see. Now let's measure this with the help of a simple scale. Let's measure. Yes, it is somewhat 2.5 centimeter. See, maybe it is not a clearer, but it is 2.5 centimeter initial height. Record this in your journal and take the pin and where the water level is present, just pin it up for the observation. Now put a simple water inside Petri plate, make it full. Yes, pour it, and now the setup is ready. We have to put this solution undisturbed for a while. See, this is how it is looking at the initial. You can see the water level, the setup is ready. Let it remain undisturbed so that endoosmosis can happen. After 30 minutes, observation. Observe that the water level is increased. We can observe very clearly. See, now let's take out this pin, the needle, and let's measure again the measurement with the stirrer. You can use any other object. I am putting inside this stirrer and I am going to measure it once again. This is our final reading after 30 minutes. That is called final reading. Now it is coming somewhere here, but it is not clear. So I am going to take my thumb and let me take the measurement properly. Yes. Okay, it is taken, and now measure it with the scale. Take the scale and again try to measure. This is our final volume. Now it is clearer. You can see this. It's a 4 centimeter increase. If you remember, the starting was 2.5 centimeter and this is 4 centimeter. Let's do the math. Okay, where initial minus final, that give you the main endoosmosis ratio. 2.5 centimeter minus 4 centimeter final, that gives us 1.5 centimeter. Now you think how this happens. This is exoosmosis practical that you have to do by yourself. This is exoosmosis practical performed by yourself. Put NaCl inside the Petri dish and simple water into potato cavity. As you can see, due to exoosmosis, water will comes out from the cell, where the potato cavity water level will get decreased, and measure the same and record it inside your journal and discuss with your teacher. Conclusion: water will move according to its concentration gradient from its high concentration to its low concentration. Thus, water level in the potato osmometer increases or decreases. Thanks for watching.